So I'm not going to be talking about neuroendocrine prostate cancer today, but I'm going to be talking about CDK12 mutated prostate cancer, which is a topic near and dear to my heart. And I also remember very clearly over when I was a first, second year fellow in Chuck's clinic, seeing a string of patients at the time with him who had CDK12 mutated prostate cancer. And Chuck turned to me and said, "John, what do you think this mutation is doing? And can you go into the lab and figure it out?" And so I've spent I spent 5-6 years in Felix Fang and Alan Ashworth's lab thinking about CDK12, and now as I've started my own lab, we're still continuing to think about CDK12. So one of the things I wanted to mention is that these mutations in cyclin-dependent kinase 12 are enriched in advanced CRPC. So when Melissa Reimers was actually a fellow, when we were both fellows, we actually published one of the first studies kind of describing the clinical features of these patients. So you can see that in advanced CRPC, it's about 7% of cases, and that the majority of patients who present with de novo primary disease have Gleason 8 or higher disease. So 90% of these patients have very high risk disease. And if you take a look at one surrogate clinical outcome that I'm showing you here, time to metastasis, you can see that the CDK12 population metastasizes at about 30 months almost faster than some of these other mutations that we typically think of as high risk. So if we take a look at how these patients do to standard of care therapy, just comparing to the BRCA mutations, you can see that these mutations do much, much poorly than BRCA mutated patients. And this was important for us to establish because at the time there was a hypothesis that CDK12 loss would generate essentially an equivalent, a functionally equivalent BRCA loss. And we now, I think, really understand that that's not the case. And taking a look at Dr. Sardar's data that he published last year in Journal of Nuclear Medicine, looking at how do these patients respond to Pluvicto, you can see again that the response rate to if you have a CDK12 mutation is only 3.6%. So what does CDK12 actually do? Well, it's a cyclin-dependent kinase that instead of regulating the cell cycle, actually regulates stages of transcription. And so you can see that, when you start transcription, there's a pre-initiation complex that needs to form. The RNA polymerase is first paused and then is released by CDK9. And then CDK12 and its really closely related cousin, CDK13, are responsible for elongation. So keeping the RNA polymerase on the DNA so you can get through the gene body and transcribe through introns and exons, and also important for termination. So one of the one of the ways that we wanted to try to figure out how can we better target CDK12 was to first generate the first CRISPR-engineered cell lines where we mutated CDK12. You can see that in panel A and B. We did this in LNCAP and C42B cells. And then we did a small molecule drug screen of over 1,800 FDA-approved compounds. And what we were really interested in was this bottom right-hand corner over here in panel C, which are compounds that would inhibit the knockout, but not kill the isogenic controls. And when we did this and subclassified the different drugs that we found two that ranked very highly, two PARP inhibitors, olaparib and rucaparib. And so because of the controversy at the time of whether or not these patients really respond or not, we chose to really show that these patients, or these tumors, these cell lines that we generated are actually quite sensitive to PARP inhibitor. So you can see both in the LNCAP and the C42Bs, the different knockout clones, we can shift the dose response very nicely to the left. And then also using another type of assay, clonogenic assay, we can see that these cells are exquisitely sensitive. Now, the interesting thing about CDK12 is that there's patients with biallelic and monoallelic losses. And so one of the things that we did in the process of generating and screening for clones that had the mutation of interest was that we actually generated a number of heterozygous cell lines. Now, what you can see here, which is really important, is that if you have residual CDK12 protein in these two heterozygous cell lines that I'm showing you, that you actually do not have any sensitivity to PARP inhibitor. And I think this is important as we think about how we interpret the results of the various trials that we've lumped CDK12 in with the HR deficient phenotype, how we actually interpret those responses. The other interesting thing is that if you take a look at the cell lines that have been chronically deleted in CDK12, contrary to what we believed initially, was that there was no difference in BRCA1, BRCA2, and many of these other HR genes. And so there's no, homologous recombination defect per se in these cell lines. And this is in contrast to when you acutely lose CDK12, that's when you actually see these HR defects. And this has now been described by Pete Nelson's group as well in a paper that's on preprint on bioRxiv. The other thing that we wanted to look at was CDK12 binds a cyclin K, which is its cognate cyclin partner, and then also again has this very closely related cousin CDK13. So we also generated knockouts and knockdowns of cyclin K and CDK13 and showed that if you get rid of cyclin K, you get PARP sensitivity, but not when you get rid of cyclin, when you get rid of cyclin K, you get PARP sensitivity, but not when you get rid of CDK13, suggesting that these are really different CDKs in terms of regulating this particular response. So we also took a look at the point mutations that are found. So the majority of patients have frame shift mutations that generate an early stop codons, what we call truncation mutations. But there's a subset of patients, about 10 to 15% of patients, that have mutations in the kinase domain that are predicted to be loss of function mutations. We generated these mutations, and one of the interesting things that we wanted to know was what is their PARP sensitivity? And what you can see here on the dose response graph is that the gold and the green lines are the two mutations that are sort of these hotspot mutations. You can see they actually have partial sensitivity compared to the complete knockout, which is here in blue. So these kinase mutations, even though they are loss of function, are not the same as when you get rid of the entire protein. And when we do generate some of these truncation mutations, these frame shift mutations here, you can see that these actually really respond like the complete knockout. So whether you have a point mutation versus a truncation mutation is actually quite important. And so we actually reinterrogated the Triton-2 data. This was a study of rucaparib in patients who had been post-ASI and post-docetaxel. And what we did is we took the CDK12 cohort and broke them up by biallelic, monoallelic, or these gene rearrangement mutations. What you can see is that the majority of patients had biallelic mutations, like we expected, about 70 to 75% of those patients. And if you now take a look at the PSA responses only in the patients with biallelic mutations, over half of them actually had PSA 30 or better responses. I'm showing you here in panel C and D. In C is a patient what their PSA was who had a PSA 50 response, and you can see that they were on study for about 35 weeks. And then here's an example of patient 2 who did not achieve a PSA 50 response, who had a PSA 30 response. But what you can see is that this patient had a fairly prolonged response on PARP inhibitor monotherapy for about 40 weeks before progression. And again, if you take a look at the by PSA response in green, what the mutations are, you can see that the top mutations are all patients who had frame shift mutations compared to patients who had those kinase domain mutations sort of in the middle. So in work that I don't have time to talk about, but in collaboration with Jean Tian and Arul Chanayan's group at the University of Michigan, we've also been looking at inhibiting the cousin of CDK12, CDK13, as another synthetic lethal strategy. What Jean did here was take knockout mice that she generated and inhibited CDK13, and you can see very nicely the inhibition of growth. And then the Chanayan lab, in collaboration with Ke Ding's lab, have also generated a CDK13 inhibitor. And you can see on the left-hand side over here, in a CDK mutant PDX, you get great growth inhibition compared to a CDK12 intact PDX. So in summary, what I hope to have shown you today is that this particular subclass, CDK12 mutated prostate cancer, is an aggressive subtype and is resistant to most of our FDA-approved therapies. And so we've used multiple approaches, including a chemical screening approach, to identify PARP inhibitors as well as CDK13 inhibitors as potential therapeutic strategies, and that the type and allelic frequency of the mutation affects response to PARP therapy and some of these different mutations that I mentioned earlier, and that we really need to be studying these particular other ways of killing these CDK12 mutated prostate cancers, either alone or in combination in future studies. So with that, thank you to my collaborators, my mentors, Felix and Alan. And also, I didn't have time to show you, but with Li-Xing Yang at the University of Chicago, we just recently have a paper up on Nature Cancer showing that the origins of these tandem duplications is really from these replication transcription collisions that I'd be happy to talk more about later. Thank you.